Welcome students. This lecture is in continuation to the last lecture on sterilization. In the last lecture, we have explained what is sterilization and why we should go for sterilization. What is the importance of sterilization in our life? Now, in continuation to that topic, today's discussion is about how many types of sterilization are there. Basically, there are two types of sterilization, mainly the physical sterilization and chemical sterilization. Now, physical sterilization includes heat, radiation, and filtration, and chemical sterilization includes use of different chemicals to disinfectant or reduce the level of contamination. The chemical sterilization includes alcohols and aldehydes, phenols and halogens, oxidizing agents and salts, surface active agents and ethylene oxide gas, dyes and vapor phase disinfectants. Basically, in chemical sterilization, we are using different chemical products to decrease the level of contamination in a particular place. Now, coming to the efficacy of disinfection or sterilization. Sorry. Basically, in this section, what we'll be dealing is what is the factors which is responsible for the efficiency of a particular sterilization. Like the efficiency mainly depends upon contact time, physicochemical environment, that is mainly the pH, the presence of organic material. That means if some lively organisms like human body or a particular cell is present, the efficiency of sterilization changes. Then temperature is also very much responsible, because by increasing temperature you can kill some microorganisms. Type of microorganism, that means which type of microorganism you are treating. Suppose some organisms are very much tolerant to high temperature, some can be killed easily at lower temperature. Then material composition is also very important, because some materials are very thermolabile and you cannot treat those organisms with, or the composition with some, with high temperature. So the efficiency of sterilization mainly depends upon all these factors. Now moving on to the next thing, that is uses of sterilization. Why we should go for sterilization? Sterilization is, this thing I have already explained in the previous slides, but then also I'm explaining in it in detail. Actually, sterilization of sorry, instrument is very necessary due to the surgical and diagnostic procedures, media reagents used in microbiology, because all these things needs to be sterilized. Food and drug is also needs to be sterilized, because if they are not sterilized, they will contaminate the environment as well as human beings. But to sterilize all instruments that penetrate soft tissue and bone, that means all instrument which is coming in contact with the body or the human being need to be sterilized. Which the instruments which does not comes in contact or penetrate, that may not be sterilized. That means if it is not coming in contact with the body, that instruments sometimes can be left out of sterilization. But which is coming in contact with oral tissue, that means suppose you are swallowing or engulfing some probes into your mouth, you need to sterilize that probes immediately, because the microorganism present in the probe will move on to another patient. In this way the things can get contaminated. So if the sterilization procedure may damage the instrument, so one thing you have to take care, that sterilization process should never damage the instrument. It should not destroy the property of the instruments. Now, uh, moving on to the ideal sterilization process, that means which processes can be called as an ideal process. That means highly efficacious. That means the efficiency of the sterilization should be high. The process should be fast, and good penetrability means the sterilization chemical used should the sterilization chemical used can penetrate the surfaces, compatible with all materials. That means the com- compatibility should be with all the materials, and non-toxic, that means it should not be toxic to the particular human beings. Then effective despite presence of organic materials, and difficult to make significant mistakes process. That means you need to monitor the process, and also you need to understand what things are going on. So all these factors are important for considering the ideality or the importance of a particular sterilization process. Now, coming to the physical methods, how to sterilize. Actually, depending upon the, as I have stated earlier, depending upon the type of materials, it will, the type of sterilization will change. Suppose the inoculating wads and loops. These wads and loops are basically made up of metals or non-metals. Okay, so this type of materials mainly requires the red heat. You need to hit the rod. Uh, those people who have worked in microbiology labs have seen that the inoculating loops before using into the culture medium are red heated. That means when red heat is occurring, then the wads and loops are getting sterilized. Now, glasswares like syringe, petri dish, test tube, all these glasswares are mainly heated in hot air oven. Then disposable syringe and other disposable items. That means these disposable items are mainly sterilized by gamma radiation. This is a type of radiation, that means it, which is very much necessary for your this is basically a chemical type of a sterilization. Now coming to the culture media contains many type of materials. So this culture media has to be sterilized only by autoclaving. Sorry, culture media containing serum and egg is sterilization. That means serum and egg is very much thermolabile. So to sterilize this particular culture media, you need to sterilize the materials. Then toxin, serum, sugar, and antibiotic solutions. These are done mainly through filtration, because if you heat all these substances, all will degrade. So you need to figure out some other possible ways of filtration. Then cytoscope and endoscope. Suppose this type of instruments are, these two are instruments. So this type of instruments are mainly your sterilized by glutaraldehyde. This is one type of chemicals which is responsible for sterilization. Then infected soil dressings in incineration. That means some soiled materials can be uh, sterilized by incineration. Skin, nowadays due to coronavirus, we are advised to wash our hands frequently with soap and as well as alcohol or sanitizer. That means skin can be sterilized by iodine, alcohol, and as well as sanitizer and soap. Then milk, that is pasteurization. We all know that we boil the milk when we bring milk from the outside. That means that process is mainly known as pasteurization. Pasteurization kills all the bacteria present inside the milk by heating the milk. Okay, now heat related methods like dry heat sterilization. In the last lecture I mentioned about dry heat and moisture sterilization. I'm not going into the detail of it. In here, red heat, flaming, incineration, hot air oven, all these things are used. The factors which is influencing are nature of heat, temperature, characteristics of organisms, and type of materials. Now, principle is what they do, they kill the organism by denaturing the bacterial proteins that lead to the oxidative damage and finally killing of the cells. Now, red heat, flaming, see in the picture, you can see that this is the illumination probe. This is heated in the spirit lamp so that it turns red hot and the sterilization is done. So, flaming is also done. Flames are used to sterilize the glass slides, scalpels, all these are sterilized by flaming. Sunlight can also act as a important sterilizing agent because the ultraviolet rays is responsible for destroying many microbes. Naturally, the rivers and tanks, rivers, in the last line of the slide, you can see that in the rivers and tanks are getting sterilized by the ultraviolet rays. And incineration means you are heating the substance to ashes. Okay, so the name of the instrument is incinerator. Here, what animal carcasses or any important soil dressings are mainly sterilized by heating to ashes. Now, hot air oven. Now, these things I have mentioned about in the previous slide, that 160 to 170, 180. These three type of temperatures can be used for different durations. So, and these are the precautions you need to measure take, because you, as you are handling very high temperatures, so chances of getting uh, here burnt up is very easy. So you need to provide all this consider these precautions. Now coming to the next section, that is heat related methods. Like these are moist heat methods. In the moist heat method, what happens? Pasteurization, that is mainly the milk is pasteurized by this method. Below 100 degrees Celsius, we boil the substances, and what happens in this time, the microbes present inside the milk gets destroyed. Now these are insufficiencies, these are different methods of your moist heat sterilization. Then this is the principle of pasteurization. For your standard, it is quite difficult, but for just your interest, I can say that we raise the temperature and then lower it so that the microbes present inside the milk get killed. Sorry. Then coming to the next is temperature. You can also heat the substances at 100 degrees Celsius, which is dry heat sterilization. Then comes the, this is the important part, that is the autoclave. We have already mentioned that autoclave must be done at 121 degrees Celsius at 15 psi pressure. This is an autoclave. Nowadays more advanced processes are there. See here, the medias are placed and steam is passed through it, and the pressure and temperature are maintained in autoclave. Now the transformation of design, see, before this autoclave looks like this one, but nowadays advanced type of autoclaves are also observed. Now sterilization conditions are, I have already mentioned, 121 degrees Celsius, 50 lb pressure. Then materials, rubber, culture media, and everything can be sterilized using autoclaves. Now sterilization can also done, you have to package the samples before sterilization. Textile, paper, nylon glass, all these things can be done. Now sterility controls. Sterility controls are nothing but how the sterility can be measured. Control, that means once you have autoclave, you cannot open it in the open atmosphere. Then again, there are chances of contamination. So different type of caps are used to control the sterility. Okay. Now these are type of loads. These are pretty important, but in your standard it is tough. So moving on, still then we will move on to the next thing in our next lecture. Till then, thank you, students.